All right, welcome to video 2. We are moving on. Hopefully you've completed the DNA structure and function material. So now we are on to protein synthesis. So last time, not last time, but a couple moments ago, we said that we needed to get somehow we need to get from DNA to making proteins. Those proteins are the things that are doing all the work in our bodies. The DNA is the blueprints for making those proteins. So we need to figure out how we are going to get from DNA to a protein. You need to go back up front to the Darwin table and pick up these two items right here, the protein synthesis notes and the protein synthesis practice. So the protein synthesis notes looks like this. There's a chart on the back side. There's a diagram at the bottom, so you need to pick that up. And you're going to hold off right now on picking up the protein synthesis. You're going to pick this up when we're all done with taking our notes today. So this will be the last thing. This is your homework. You need to complete this. So let's get into our notes. All right, so protein synthesis notes. We are going to do a quick recap here. Let's go into presenter mode. All right, so we know that genetics is a study of how traits are passed from parents to the offspring. Those traits are specific characteristics like height, coloration of your skin, color of your hair, color of your eyes, length of your toes, all of those things. Well, here's a wonderful thing. Proteins are made to give you those characteristics and make you who you are. So we need to remember that traits are determined by the genes. That is a specific section in your DNA. Remember that DNA makes up your chromosomes, and that genetic information is passed from parents to the offspring. One gene controls one trait. Now, I'm going to be honest with you, that's not always the case. We're going to just say for the sake of this class, we're going to say that one gene controls a trait. There are some traits that are a little bit more complex than that, but we're going to stick with that thought for now. So the DNA has that genetic information, and it's used to build a protein, and proteins run all of those living things. So here's a recap from way long ago. This is not an egg in a frying pan, but this is representing our nucleus. We know that in our nucleus, we have DNA, and that DNA is locked away in the nucleus. We know we want to protect our DNA because if damage is done to it, that wouldn't be good for us. So we want to protect it in the nucleus. We know that proteins are important, and we know that proteins are going to be produced by ribosomes, either found on a rough endoplasmic reticulum or free-floating in the cytoplasm. We also need to go back and look at the fact that proteins are made of these small molecules called amino acids. So that's from way back when. We know that amino acids are used to build proteins. So somehow we need to get from inside the nucleus here to outside the nucleus, somewhere out in the cytoplasm or the rough ER. So if DNA is stuck in the nucleus and the ribosomes are in the cytoplasm, we need to figure out how we're going to get to that point. How are we going to do that? Well, there's a couple different ways that we could maybe do that. If you have something that's locked in one place and you need to get it somewhere else, nowadays we just whip out our cell phone, we take a picture, and then we can share with the world on Twitter or Instagram or Snapchat or Facebook, or maybe even you update your MySpace page. Yes, all of you probably, well, no, probably none of you have a MySpace page. But anyways, so we need to get that information from the nucleus. You're taking that picture, and we need to get it out to the world. So here's what we're going to do. We're going to figure out how we do that. Well, we have James Watson here. He determined that DNA. Remember I said that maybe he sort of stole someone's work, but we can talk about that conspiracy theory later again on Thursday. We need a messenger. And nowadays your message is usually in picture form. So we're going to kind of use that analogy as we go through this. So we need to get that message out to the world, and we're going to figure out how we do that. So your learning targets, please fill these in at the top of your note page. So you can pause and fill that out. I'm going to move along here. So if you need to pause, go ahead and do so, but here we go. So question 1, what is the messenger? Well, we have this awesome thing called messenger RNA. Now, you need to specifically call it mRNA because we have a couple different types of RNA that we're going to be working with. mRNA stands for messenger RNA. We are going to carry that message. Whoop, there's those sounds again. Nope, let's get rid of that. So we are going to carry that message from the nucleus, from the chromosomes and the DNA in the nucleus out to the cytoplasm to meet up with the ribosomes. We are going to build proteins using that message so that our bodies can function and do everything that they need to do. So this is our RNA, specifically mRNA. Now, RNA structure is a little bit different, but still similar to DNA. So we said that DNA was deoxyribonucleic acid. Well, now we have ribonucleic acid, which is RNA. The differences we're going to see is that RNA is a single strand. It is not a double-sided. We can see comparison right here. So we have DNA is a double-stranded. It looks like a spiral staircase. And RNA looks like a spiral staircase that you cut in half. It's only one side of the staircase. There is a slight difference here, and I'm going to move my picture over so that you can see this. So we have 4 bases. Before we had C, G, A, and T, but now when we're making mRNA, we are going to use C, G, A, and U. So that replaces thymine. When we are making RNA, so we use DNA as our template. So we're saying, okay, we're looking at DNA. Here's our template. We are going to make an RNA molecule from that DNA template. That's called transcription. Back in the day when they had scribes, and what did scribes do? They wrote down things that the king said and recorded things on paper. So that is transcription. So to transcribe something is to write something down or to copy it. So transcription, the process of transcription, the first thing is the DNA has to unzip. It's like a big zipper and it's got 2 sides, but we need to be able to read these bases that are here, are nitrogen bases. You notice that the T isn't here right now because this is a DNA molecule. We're going to unzip it and we're going to use this side right here as our template. Now, during transcription, we are going to match RNA bases to DNA bases on one of the DNA strands, and we're going to use T as the partner instead of, we're going to use U instead of T if there is an A. So you can take a look at our strand here. We had a T here, so we had an A, and we can work our way through this. We can practice. So I want you to pause this and look at it and see if you can figure out what the mRNA strand is going to be. So pause it right now and look at this strand. Figure out what is the matching RNA going to be. All right, so you've had an opportunity to look at this. Hopefully you came up with something like this. So our complementary side here, our mRNA, we have a T right here, so we have an A. We have an A on the DNA, so we have a U. If we were making more DNA, this would have been a T, but now we are replacing it with U whenever we have an A. So hopefully you got something like this, and we are going to move on. So what do we already know? Well, we know the DNA has instructions for making proteins. Those have to remain in the nucleus. They're locked away for protection. The mRNA carries the instructions for building our protein out into the rest of the cell outside of the nucleus so that we can build these proteins, and we are going to use amino acids to build that protein. Mr. Crick here is having a bad hair day. But what reads the information? So we know that we have our message, but something still needs to read that message. Well, this is where you come into play. This is where you should already know this information. We are going to use a ribosome. Yes, remember ribosomes make proteins, so ribosomes read the information carried on the mRNA to build those proteins. So we have our mRNA. This is our mRNA chain right down here. Move my picture up here. So we have mRNA, but we still need to make a protein. So we have a ribosome that is going to kind of move along the mRNA, and as it's moving along, it is going to read the mRNA and build that protein. But we need a little bit more information before we can do it. This is like one of those information, but just wait. There's still more. If you buy 15,000 of them, you get a deal. No, I know, sorry, just trying to make it a little interesting for you guys. So we are going to have to translate. Think about it. Translation is going from one language to another. So we need to go from the language of RNA into the language of proteins. So we are going to go through the translation process. So we take the mRNA to build our proteins. Here's our mRNA. So taking a look at the bottom of your diagram on the note sheet, you should see that there is a place for you to put all this together. So we have, and it's hard to see, but you can see that there's some boxes. So we need to have DNA, and we need to make mRNA. We are going to make that through the process of transcription. So you can fill that in. And then we are going to take the mRNA and we're going to translate it using a ribosome into a protein. We're going to use a bunch of amino acids to build that protein, and then that protein becomes this awesome thing. It becomes the trait, and it makes you who you are. That's the process of going from DNA to a protein. So, recap. mRNA has instructions for building proteins. Proteins are built by ribosomes. There are these long chains of different amino acids. The ribosome reads the mRNA and adds amino acids to build the proteins. But we have one other thing we still need. What is bringing those amino acids to the ribosome? We need something. They're not just going to be free-floating and guess. This is going to be a very specific project for a molecule. And that molecule is transfer RNA or tRNA. So tRNA, and you can see this right here. Here is the tRNA molecule with all those amino acids attached. It's bringing them in, dropping them off. Here's our mRNA, and you can see how it fits like a puzzle. If you have ACC, on this side you're going to have UGG, and that tRNA brings in the correct amino acids. And as it goes along, it keeps on adding and adding and adding and adding and adding and adding and adding until you get your protein. So, another recap. Whew, lots of recaps. So, DNA stuck in the nucleus. We need transcription to make mRNA. Done. We are going to take that mRNA and we are going to translate it into our protein factory, the ribosome. The ribosome factory is going to need a little bit of delivery to help it out. So we have these tRNAs that are going to bring in the amino acids to help build that protein. And in the end, all those amino acids are going to build or be put together to build our trait. Now, here's the thing. There is a chart on your note sheet. This is where it's going to be a little bit tricky because I worked with the other class. I'm not there to help you. So you need to be able to do this on your own. And I know it's going to be a little tricky. You have the table. You are going to utilize the table that is on the very back side, the very last thing on your note sheet. You're going to read it and you're going to look at in sequence the first base. It says on the left side. The first base is what letter? It is going to be that row. Here, let me pause the video for a second. So you are going to read your mRNA in a certain order, as I was saying. You have to read the first base. So look on this left-hand side here. What letter does the chain start with? So you look at what row does it start with? So it's going to be, if it starts with a U, what you're going to look for your amino acid is going to be in this first row. If it starts with a C, it's going to be in this row, A, G, so on and so forth. Then you look for your second base in the order. And you go, okay, well, if it was C and the second letter is C, now it's going to be in this row and in this column. And if the last one is A, CCA is going to be the amino acid pro. You just need to know the abbreviations for now. That's all you need. Okay? So you are going to take all of this information and you are going to hopefully be able to figure this out. So this is this is how you're going to take this. You're going to read them 3 at a time. So I read AUG. AUG on your chart codes for MET. CGU codes for ARG. You just go down. So you chunk it into sections of 3. The ribosome is going to read that. You're going to chunk it into triplets, and those codons, you break it into the codons, so you have sets of 3, and then you take your chart and you read it and you figure out what amino acid correlates with those bases. And as you can see, all of these code for it, so those are codons. That's the last part of your notes. So what you're going to need to do right now is you are going to use this information. You're going to go up and you're going to grab your protein synthesis practice and you're going to complete that. I know there's going to be questions. I will be there on Thursday to help you guys work through this. I know this was not the most optimal situation with me being gone today and we having a snow day on Friday, but it is what it is. You're going to work through it. You guys are troopers and you're going to be great. So I will be there on Thursday to help you. One last time, we're going to go through this, blah, 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 blah. So you are going to complete the practice. Keep in mind, that I'm out of time. Bye.